Consider this. You are starting a family, but you have the opportunity to choose the traits of your future children. What would you do? Well, this might sound like science fiction for now, but in the near future, this might actually be a reality. Hi, my name is Tommy Renshaw. I'm a junior at Howard High School, and I am enrolled in the independent research course in which I am studying CRISPR, its concerns, and its controversies. Many people have heard of CRISPR but not know how it actually works. Well, CRISPR is a recently discovered technology that allows scientists to edit the DNA of any living organism. DNA is basically the code that tells your body and its cells what to do, and tells the cells of basically any living organism what to do and how to recreate and how to function. CRISPR is basically derived from a protein of a bacteria that allowed it to edit the DNA of whatever cell it was infecting. DNA is the double-stranded version of this code, while RNA is the single-stranded version. And when a single strand is connected to another strand, it makes the double helix that we're all familiar with. Now, scientists use this single strand, the RNA, by attaching it to the custom designed RNA that they wanted to insert into the DNA. Basically, they use the guide RNA that matches the targeted DNA to guide the CRISPR protein to the DNA. Then you see the CRISPR protein literally cuts the DNA into a single strand and then attaches the custom sequence that it wants to have to it. This allows scientists to edit, delete, or modify any gene that they want to. This technology obviously has huge potential. First of all, it can treat any hereditary disease such as sickle cell, Alzheimer's, some sorts of hereditary blindness, basically any disease that can be passed down from ancestor to descendant through their genome, or just any disease that has anything to do with genetic mutations, such as Alzheimer's, some sorts of cancers, and many other diseases like Huntington's as well. In addition, because of its ability to edit the genes of plants, scientists can create plants with more favorable traits that allow them to either grow better, grow faster, resist pesticides or insects, or maybe even have more nutritional value. Lastly, because of the guide RNA function that the CRISPR protein has, it could diagnose certain diseases more effectively and more accurately than any other technology that we have currently. However, there are also concerns that stem from this uh, technology. Most notably is the commercialism of CRISPR. Now, commercialism entails uh, companies using this technology for commercial and monetary gain. This can be done by companies patenting certain treatments or technologies that involve CRISPR. This means that these companies can basically restrict who can access these technologies and these treatments. They might they might be able to restrict it so that only those who are wealthy can afford these treatments or these technologies. This could lead to wide social inequity, as those wealthy who can afford these treatments might possibly eradicate these diseases that are treated using CRISPR entirely from their genome, while those who are poor and who can't afford or uh, gain access to these treatments might continue to live with these diseases and with these unfavorable illnesses. In addition, this could also be equated to farmers or agriculture, as if they don't have access, if they can't pay for this new uh, technology that allows them to grow better crops, then they simply have a disadvantage. On top of all of those concerns, we also have ethical concerns that stem from the use of CRISPR. Specifically, we have human embryo engineering. Human embryo engineering is controversial because embryos cannot give their consent to be edited, and when we edit humans, we often want the consent of the subjects, and obviously human embryos cannot give their consent. Also, when human embryos are edited, uh, the genes that are edited could be passed down to the embryo's offspring, their entire germline. This obviously is a concern because if a mistake is made, or if an unfavorable gene is inserted into the embryo's genome, this could affect many people more than the researchers would have known would have liked, or more than the researchers could even know, especially since the effects of those genes might not even show up until the embryo gives birth to offspring or later down their germline. Now, this picture of He Zhuangqi on the right encapsulates this concern because in 2018, he actually announced that he gave birth to two embryos that were twins that had genes that allowed them to be resistant to HIV infection. This was controversial because there are other ways to prevent HIV infection or to treat it if one is infected, and therefore he put these twins and their embryos in danger, and perhaps their entire germline in danger by using technology that wasn't even fully developed to accomplish something that could have been accomplished in another way. This concerns lead to one question: how could this be stopped, and how could these problems be regulated? Well, actually, through my research, I found that there is a significant lack of regulation of CRISPR, and this actually might be the most concerning part of gene editing technology. Well, I found that there's a lack of strict guidelines. While some organizations have put out guidelines on how to use CRISPR, these guidelines are not specific and are not strict. In addition, there's no enforceable regulation. These guidelines have been put out, but they cannot be enforced. There's no specific singular body that can enforce this regulation. Hence why He Jiankui was able to do what he did. In addition, there is no scientific consensus around the technology. This makes it harder for guidelines to be reached because there's not one voice uh, in these researchers that say when and what situations CRISPR can be used for. Here on the left, you see a picture of Jennifer Doudna, who is actually one of the co-discoverers of CRISPR, speaking at a convention. These conventions about human genome editing are often where much of the discourse is had. However, there's been a lack of an actual consensus being reached in these conventions. And lastly, there's a lack of public input, whether because scientists simply are not hearing the input of the public or because the public are not educated enough on this subject. There's simply a lack of public input on their opinions on CRISPR and its potential uses. This leads me to my primary research, in which I hypothesized that when asked, both the general public and researchers would believe that while the benefits of CRISPR do outweigh the concerns, there's a need for stricter regulation. In a survey I conducted, I separated the contestants into those who were familiar with CRISPR and those who were not. Those who were familiar with CRISPR believed overwhelmingly that one of the main drawbacks of CRISPR was the unethical enhancement of humans, which would include human embryo engineering. They also believed that several other things could be a big drawback of CRISPR. When asked, they also believed that the government should play either a very heavy role or a moderately heavy role in regulating CRISPR. Those unfamiliar with CRISPR also mostly agreed that the government should play a moderate or a very heavy role. This supports my hypothesis. To conclude, CRISPR is a revolutionary technology that could have global implications both negatively and positively. While the obvious positive benefits of it medically and agriculturally suggest that it should be a technology that will be used widespread throughout the entire world, this also denotes that we need to ensure that safe and strict regulations are put in place so that equity and equality is guaranteed throughout the world.